So in the ESMO Congress, I presented the results of the Pioneer trial, which is a phase 1b/2a clinical trial designed to overcome resistance pathways to PD-1 and PD-L1 inhibitors in advanced non-small cell lung cancer. This trial is part of a large program aimed to study and overcome resistance to PD-1 and PD-L1 inhibitors in non-small cell lung cancer, with a biomarker program and then a clinical trial program. This clinical trial program results from an academic network of clinicians and researchers. We enrolled patients with advanced non-small cell lung cancer, ECOG PS01, who had disease progression after PD-1 or PD-L1 inhibitors. The patients were randomized between different combination arms with durvalumab, one arm with the NKG2A inhibitor monalizumab, another one with the oleclumab CD73 inhibitor oleclumab, another arm with the ATR inhibitor ceralasertib, and another arm with the MEK inhibitor savolitinib. And all these experimental arms were compared to the standard chemotherapy docetaxel. The primary endpoint was the 12 weeks disease control rate. In this trial, we used an innovative and adaptive design with a Bayesian approach, where we could, during the trial, stop quickly arms with no signal of efficacy, or on the contrary, add new arms when new data emerge with promising new combinations or molecules. We enrolled 114 patients in the trial. 2 arms were stopped prematurely. The arm with oleclumab was stopped after only 3 patients because of no efficacy signal reported in a concurrent trial. And the savolitinib arm was stopped because of futility shown in the first interim analysis of our trial. So finally, no experimental arm succeeded in meeting the Bayesian criteria of efficacy in comparison with docetaxel regarding the primary endpoint 12 weeks disease control rate, but also the secondary endpoints with response and survival. However, when considering individual outcome data, we saw that some patients in the experimental arms with monalizumab and ceralasertib derived some durable clinical benefit from the combinations, with significant responses in the target lesions and with long-term clinical benefit of more than 1 year. When considering the standard clinical and biological data of all these patients with durable clinical benefit, we could not see any similarity, and there is a real need to go further with biomarker analysis in order to be able to identify these patients. These biomarker analyses are ongoing. The safety profile was as expected, maybe with the exception of the ceralasertib arm, where we had to adjust the dose and schedule of ceralasertib in order to ensure a better safety and efficacy profile, and especially for safety for the hematologic toxicity. So to conclude, the Pioneer trial is an academic collaborative success with an innovative and adaptive design with a Bayesian approach, which is quite original. We were able, with this approach, to experiment several combination arms in comparison with docetaxel, whereas no experimental arm met the efficacy criteria in comparison with docetaxel. we saw that some patients had long-term clinical benefits, especially from monalizumab and ceralasertib combinations, and biomarker analyses are ongoing in order to be able to identify these patients.